So just to introduce Professor Petrovsky, he's the research director of Vaxine Pty Ltd, a professor of medicine at Flinders University and an active clinician. He's held multiple large research grants from the NIH in the US and has authored over 200 peer research papers. He's won several very prestigious awards, including an Ernst & Young Entrepreneur of the Year award in 2010. And his recent focus is the development of a protein-based vaccine against COVID-19 called Covax19, which is now approved as both a primary course and third dose booster and forms part of the Iranian National Immunization Program. So over to you, Nikolai. Thank you very much for joining us, and apologies for that mess up from myself.
No problem, Jeremy. I was falling a little bit asleep. It's about midnight, I think, here. So, but thanks for giving me the opportunity to tell you a little bit about actually the first vaccine. I've developed many vaccines. This is the first that's actually been approved for human use and been rolled out on a large scale. So it's been a very exciting time for us. But I started life 20 years or so ago in the vaccine adjuvant space. And this was our original technology for which we got funding from the US government, which is essentially a sugar-based adjuvant. It's based on Inulin, which is a polyfructose polymer with a terminal glucose, which interestingly it has no immunological activity in the soluble form. It's actually used as a health food, and often you'll find it in most yogurts and kefirs. But if you crystallize it into these donut-looking like structures, it develops a very potent adjuvant activity both on antibodies, but also on both CD4 and CD8 T-cell immunity, as I'll show you. So I think, you know, when you go to vaccine talks, most people are focused on the antigen. Obviously, you need an antigen to drive a vaccine response. But if you think about the antigen as the engine, then what we have with the adjuvant is the turbocharger, which takes, you know, a very weak and not very powerful engine and turns it into a Ferrari. So that's what our adjuvant does. It's the Ferrari maker of the vaccine world. And of course, the reason we need vaccine adjuvants for protein antigens is to enhance, you know, all manner of immune responses, whether it's antibody levels, T-cell responses, better cross presentation and cross protection against variant strains, reducing the amount of antigen, making the vaccine last longer, which might be quite relevant to COVID-19 because the vaccines are very short-lived, improving immunogenicity in the very young and the very elderly. And in fact, we're just doing a children's study right now with our COVID vaccine. And also trying to generate immunity, not just against infections, but potentially also against cancer. This is really just highlighting what the adjuvant does. This is some influenza work from many years ago. But it's just highlighting that you take a standard seasonal influenza vaccine, which we all get every year, you add this sugar-based component, and not only do you see enhanced antibody titers, you know, quite dramatically increased, but you see dramatic increases in memory CD4 T cells, and even more dramatic, you see this very large increase in the memory CD8 T cell population. And this is quite surprising given that this is a pretty inert sugar-based adjuvant component that's non-inflammatory. So over the years, with help from the NIH, we've applied this to any number of different models. A lot of these are lethal challenge models, and we've been able to show that by adding this turbocharger, you know, we can dramatically enhance protection really across the board of pretty well every adjuvant antigen that we've tried. And we've also used this in the context of allergy and cancer and even Alzheimer's disease and vaccines of drug addiction. It's been highly successful. But obviously, right now we're talking about COVID-19. So after many years of testing this in humans, in influenza vaccines and hepatitis B vaccines, in the last 2 years, we had the opportunity to really show how this works in the context of a recombinant protein-based vaccine for COVID-19. So we went through a phase 1, then a phase 2, finally a phase 3, which was undertaken in Iran in just under 17,000 subjects, randomized to either placebo or to receive our adjuvanted recombinant protein. We got approval last year in October as the first recombinant protein-based vaccine to get approved. And since then, we've also got approval now for its use as a third dose booster, and we're well underway with a pediatric study to get an extension with that. So in terms of the vaccine itself and the data, so this is the construct. So essentially it's the full length of spike protein, although we've truncated it, we've removed the transmembrane domain to make it a soluble secreted protein. So it's a bit different to Novavax. We've removed the furin cleavage site, and then we've incorporated various mutations as the different variants of the spike protein have arisen. It's a full-length 180 kilodalton protein, so it's pretty big. It's heavily glycosylated. It forms into the natural trimers. You can see these cone-line structures under negative EM. And we just formulate these with our adjuvant, and that's the final product there. So essentially, this is expressed using the baculovirus system, which most people know. It's been used by Novavax and also by Sanofi for both influenza and COVID vaccines. So we're able to basically go from the gene sequence, codon optimize, synthetic gene, splice this into the baculovirus backbone, transfect the insect cells for 3 days. They secrete the recombinant protein. We purify that and then just formulate it with the adjuvant. So it's quite a fast process. We've tested it in multiple animal models, hamsters, where we've looked not just at homologous protection, but we've also done challenges with the beta variant and also the delta variant, ferrets and elderly monkeys, and we see very consistent effects where we get complete lung protection. We also reduce nasal virus shedding across all models, and in fact, we've now got data showing we can block transmission from a vaccinated animal that's challenged to a naive animal put in the same cage. And obviously, we now have all the human data. Essentially, the phase 3 study showed against Delta variant a 60 to 65% protection against symptomatic disease, and the vaccine was approved on that basis. And then with the phase 4 booster study, we're able to show, irrespective of what vaccines people had as the primary course, whether it was an inactivated whole virus or an adenoviral vector vaccine, we got between a 10 and 25-fold increase in neutralizing antibody with the single booster dose. So this is just now going into the immunology of the response. So this is in mice, just looking at the recombinant spike protein by itself. As you can see, recombinant spike protein, not surprisingly, not that immunogenic, and certainly doesn't really induce much in the way of memory CD4 or CD8 T cells. When you formulate it with the adjuvant, you enhance the antibody response, particularly the sort of Th1 type antibodies like IgG2C, which you can see are not being induced by the protein, but you add the adjuvant, you now get very nice enhancement. And you can see dramatic increases in memory CD4 T cells, whether it's a Black 6 strain or a BALB/C, you see the same thing. And then these dramatic increases in the memory CD8 T cells in, again, both the Black 6 and the BALB/C. If we drill down and look at the cytokines, you can see that we're actually enhancing both Th1 and Th2 T cells with the adjuvant. So it's not so much a biasing as it's just enhancing both sides of the immune response. So we then did a ferret challenge study at the University of Georgia. Cut a long story short, obviously we've given 2 intramuscular doses of the vaccine, challenge 2 weeks later. We then do lung histology. We can look at the level of inflammation in the lung of the animals, and we can also stain for the virus itself in the brown stain shown here. So these are the control animals. You can see their lungs are extremely inflamed. There's very little air spaces left, and you can see high levels of virus replicating in the lungs. So these are the unimmunized animals. And then on this end, you can see the animals that have been immunized twice with the Covax vaccine, much less inflammation in the lung than you have in the control animals, and no visible or detectable virus in the lung. And then interestingly, when you look at the nasal swabs and nasal washes in these animals, what was really interesting is that we could detect virus in the control animals and in fact in single immunized animals, but the animals that have had the 2 doses, there was no detectable shedding even at day 3, which is very early. And we see the same thing in the hamster model. So I've already introduced the phase 3 study. This is looking now at cross-neutralization. So these are samples from the people in the phase 2 study looking at their blood 2 weeks after the second immunization and looking at, with a lentivirus pseudo-neutralization assay, looking at their ability not just to neutralize the original Wuhan strain, which is what the spike protein was based on, but also across all of the other variant strains. We do now have the data for Omicron as well. What you can see is definitely that there's I guess, very little loss of activity across Wuhan alpha and beta strains. You do see a bit of a step down by about 2 to 3-fold to gamma and delta. And then there's about a 2-fold, as I say, it's not on the slide, but there's about a 2-fold drop down to Omicron. But overall, we're actually seeing very good cross neutralization across all of the variants, which is obviously very reassuring. In terms of vaccine safety, which is obviously very important, we see very little in the way of side effects. There's a bit of a sore arm, which is, again, very typical of an adjuvanted protein-based vaccine. Some fatigue and headache and myalgia in the first day post-vaccination. Not a lot in the way of fever or severe side effects. And certainly we haven't seen any thrombosis, and we haven't seen any myocarditis. And I think we're now up to 6 million people immunized, and we haven't seen cases of those side effects, which is very reassuring. In essence, these are the ongoing clinical studies. As I said, we were approved for adult use. We're not yet approved for pediatric use. Hopefully that will come with the pediatric data over the next few months. We're doing some chronic disease studies and also looking at the dose interval. We use a 3-week dose interval, which is quite short. So we're looking at what happens if we extend that. And needless to say, we also have been looking at, do we need a vaccine specifically targeting the variants and particularly Omicron, although with the booster study after 3 doses of the Wuhan strain of the spike protein, we actually have very high titer neutralization of Omicron. So like the other vaccines, interestingly, 3 doses does give you protection against Omicron. So obviously, everyone's talked about mRNA and adenoviral vectors. I think it's now the time for the proteins. Obviously, there's Novavax has been approved. We've been approved now. Sanofi's coming through. So I think, the next year or so, we'll be hearing a lot more about the benefits of the proteins as opposed to the other platforms. And of course, I think safety is really the thing that I would hang our hat on because we haven't seen any of the side effects that have been reported with the other platforms. Obviously, there's a number of advantages to the protein-based platform, particularly the temperature stability, low cost of production, and also the speed with which we can make new proteins. So I think just to finish, just to acknowledge all the partnership with Synergen in Iran, which has enabled us to get through the phase 2 and phase 3 studies and get this onto the market. We're looking at getting it approved now in a whole range of other countries. All of our academic and also obviously all of our funding and industry partners, which we couldn't have done this project without. So thank you, and happy to take any questions.
Thank you very much, Nikolai. That's great. And again, apologies from my side from the very beginning and forgetting about you. That's my very, very big mistake. Anyway, we do have a question. Our first question is from Olivia Stricker, asks, I'm curious if the adjuvant mechanism is understood. Does it act as a lectin? That's a very good question. We've spent 20 years trying to decipher how the adjuvant works. We know it's anti-inflammatory. It actually suppresses interleukin-1 and most of the TLR pathways. So we do think, yes, it is a sugar, and so we do believe it's probably interacting with some of the C-type lectins. That's as far as we've narrowed it down, but we haven't found, if there is a single receptor, we haven't yet identified that. But we think it's in the family of the C-type leptins, and maybe it's multiple of those, not just one. Great, thank you.